This is essay or unit 2 essay question 1. So in each muscle, perpendicular to the long axis, every single muscle is built of what you call a sarcomere. So we're going to draw a sarcomere here, and then we'll talk about its structures. So each sarcomere is built of a few different pieces. The first piece, let me get it drawn out here, are these here. These are called the Z discs on both sides, and they are separated in the middle by the M line. That M line is present in all sarcomeres, and it's just kind of the division between the two edges of these sarcomeres. The sarcomere also has, we'll draw it in a different color here, what we call titin, and it's between all of these, which I'll describe in 1 second. After I draw out this titin, and then attached to this titin is another filament here called myosin. So green is myosin, and blue is the titin. Attached to the myosin are myosin heads that come off like this on each one, and there's many of them, and these are part of the contractile filaments. So we'll see, orange here is the myosin head, and then drawn in black, and I'll draw over it with a little bit of gray, kind of wrapped around small in it on all of these. So the black is what we call the nebulin, and then wrapped around the nebulin is called the actin, and that surrounds all of these. So the gray is the actin, and that actin surrounds all of the nebulin. These individual are the G actin, so individual is G, and the group that's wrapped around in the helix is F actin. So that is what is a sarcomere is built out of. The myosin heads, or the myosin and myosin heads, so we'll draw a string of myosin here. It's kind of wrapped around itself. And this is called the tail of the myosin. And then those myosin heads, they come out like this, and they wrap around on either side. And then they have this section is the neck, the hinge neck of the myosin. Also, it's there. The actin, we'll blow up one of these actins. So I also forgot to write this black is the nebulin. So we'll draw a bigger section here of nebulin. We'll cover this in actin. We'll draw it in 2 different colors. So these individual, like I said earlier, are the G actin, and then as a group, it's the F actin. And they're wrapped around this nebulin in a helix shape. So this will just be one strand of it, and then we'll draw one more strand in another color to show you the difference. So on each one of these strands is what we call, draw it in purple, is what we call tropomyosin, and it binds to the active binding site on these actin. So it goes here, and it blocks the myosin heads from interacting with actin. The actin or the myosin heads need to bind in order for muscle contraction to occur, and so this tropomyosin is in the way. So we draw it with a structure here called troponin. Troponin kind of looks like a Mickey Mouse head here, and there's 3 globulin particles to it. So the first is troponin I, so troponin I, and then we have troponin T, and then we have troponin C. So troponin I is what binds to the tropomyosin here. So it would bind like this. So this is I. This would be T, and this would be the troponin C. As this troponin I binds to the tropomyosin, it moves it out of the way and allows myosin to bind. The troponin T is what will grab onto that tropomyosin and move it. And then this here, troponin C, has 4 binding sites. Let me write that out for you. Has 4 binding sites. And 2 are high affinity and 2 are low for calcium. And that is a part of muscle contraction. The next thing that I want to talk about is what happens as an action potential reaches the neuromuscular junction, and how that will result in the release of calcium from the sarcoplasmic reticulum, or the SR. So let me draw here a neuromuscular junction. So here we have it reaching. Here there would be calcium channel. And then here we'll have some nicotinic 1 receptors, and then some sodium channels as well flowing that way. And this calcium entering will release vesicles full of neurotransmitters, full of acetylcholine into this, which will activate the nicotinic 1 receptors. This will give us a graded potential. So if we draw an action potential graph here, we'll start to see it moving towards threshold, but it's not going to be enough. And we're not going to reach that threshold for the full action potential. But as it hits, it's depolarizing and causing these charges to move towards what we call the T tubules. These T tubules are just long and thin tubes that extend out from the neuromuscular junction, and adjacent to them is the sarcoplasmic reticulum, which I'll draw here. And it basically folds all the way around all of these structures. So, and it's on both sides, it's everywhere. Let's draw 1 tube coming out of it. And this is full of calcium. We'll draw it here in red. Calcium, all over the place. And there is one protein called calquestrin that keeps all this calcium right against the T tubules. So when we have the action that I'm about to show you, that we for sure have it moving down its concentration gradient. So if I were to blow one of these up, this T tubule here, just make it really small right there. There is 2 proteins in here. One is called dihydropyridine, and it's called a channel, but it doesn't actually open like a channel does. It actually is basically a reception channel for this structure I'm drawing now, which we call ryanodine. So this is, we will call it R, Y, R, and D, H, P, dihydropyridine. So when these charges come down for the depolarization, it activates this DHP to then open the RYR, the ryanodine. So as this opens, and this is in blue right here, I'll draw the sarcoplasmic reticulum all along here. And so there's calcium. These red dots are all calcium here. As this opens, let me draw one just below it, another open one. So we'll have the DHP here, but we'll have an open section of the RYR, the ryanodine. And so this calcium will start to rush out from the SR. And so there'll be calcium flooding into the ICF, the intracellular fluid. And as that is happening, there is circuit pumps that line the SR. They would be just along the SR here. All these green boxes are circuit pumps, and it takes this and pumps it right back into the SR to be used again. After we get that calcium to move into the cell, we go through what we call the cross-bridge cycling. So I'll show this using half of a sarcomere. So we'll show it with the myosin and how it attaches. So we'll draw out here the actin that it attaches to. And so we're just have it drawn half here like this. This dotted line would represent the other half, but we're not gonna use the other side. Just it's happening simultaneously on the other side. So this is the actin that the myosin is trying to bind to. And we have here what we call a cocked myosin head with molecule ADP here and a phosphate attached. What happens is we'll call this is going to be step 1. And so what we just went through, we have calcium that enters the cell here. As calcium enters this cell, the myosin head that still has the ADP and phosphate attached to it will then attach to the actin. Now that it is attached to the actin, there's heat that is released from the muscle, or from the actin body. We'll call this step 2. Step 3 is an important step because we have two things that fall off. We have the ADP that falls off, and we have the phosphate that falls off. And then the myosin head does what we call, it will uncock. So it is no longer cocked, and that looks kind of like this. So there's no longer ADP in the myosin head, there's no longer a phosphate attached. This is step 3, and we'll call this the power stroke. This is where that muscle has contracted. And then in step 4, we finally have a molecule of ATP that returns, and it will drop it on there. It will it does not recock the myosin head. It still is uncocked, but now we have a molecule of ATP here. Sorry, that was poorly drawn. And heat is still released. It no longer is attached. There now is a, I'll draw gray. There's a space here. It has come off. And then it will cycle all the way back to step 1 in a step called hydrolysis. And because that calcium here in the step we talked about previously had entered into the ICF, this whole step happens, and it's what causes the actual muscle contraction to occur. The last things that I want to talk about are the energy sources and how they're utilized in muscle contraction. So we'll go through the 4 different types and kind of talk about them, some of their advantages and disadvantages, and how they're used. So the first is cytosolic ATP use. It's good for approximately only 5 seconds. So it's about 1 to 5 seconds. This is mostly used just for cellular processes that are happening. And so it's not a great use of the energy that we have. The second is the creatine phosphate. And this is good for approximately 5 to 10 seconds. So not much longer. So let me write this down. This is like for cell processes. A good use of creatine phosphate is when we're weightlifting and you need a quick 10-second burst of high intensity energy and you can lift a heavy weight, but it is still ineffective. It's similar in the sense where you take an ADP plus an inorganic phosphate to make ATP. In this case, we're taking an ADP and a creatine phosphate to make ATP, and they use creatine kinase, which is a better use than what the cytosolic is, where it's just the ADP to ATP. The third process we can use is called glycolysis. And this is where we take glycogen to glucose to ATP. And this is good for about 60 seconds or about a minute. So a little bit more energy, but still not great. It's poor because it just doesn't give us much. For every molecule of glucose, we only get 2 molecules of ATP. So it's just an inefficient process. That's inefficient process. And then the fourth step, or the fourth option, is just called aerobic respiration, RESP. And this can last you for hours. It's not technically indefinite, but it can last you for a very long period of time. So these first 3 are what are known as anaerobic. They're anaerobic. And then this last one obviously is aerobic. So it uses oxygen to give us the energy that we need. They are all serve good purposes. Aerobic respiration is great for just kind of going throughout your day. Like if you're going on a walk, aerobic respiration is great. You take in a lot of oxygen that gives you plenty of energy to be able to continue on with that walk, and kind of other more simple parts of your daily life use aerobic respiration, which is very helpful, that we don't need to use one of the other processes to do our basic tasks. Otherwise, we wouldn't be able to do them for very long. But those are the four types that we have that help us with our muscle contraction to use our bodies.